Hello, students. A very good afternoon to all of you. So today we'll start the topic from restriction enzymes from the topic principles of biotechnology and processes. So in the last class, we were talking about restriction enzymes, especially when it comes to the tools of recombinant DNA technology. So when we discussed about restriction enzyme, I told you that they are the class of nucleases, and discovery of restriction enzyme itself was one of the most amazing factor when it comes to biology. So when we talk about restriction enzymes, we can categorize them based on their activity into two classes. As I said, one is exonucleases, other one is endonucleases. So when we talk about exonucleases are the class of enzymes. Those, for example, if you have a long chain of DNA, they have the capability to recognize only the ends. Okay, they cut only at the ends of these genes or the DNA. So these are called as exonucleases, and their application when it comes to the recombinant DNA technology is highly limited. So we don't use exonucleases basically. So when it comes to endonucleases, restriction endonucleases are of three types, basically type 1, type 2, and type 3 restriction enzymes. Okay, within endonucleases I'm talking about. So when we talk about the three types of endonucleases, and we use majoritarily type 2. In all cases, we use type 2 restriction enzyme only. So why don't we use type 1 and type 2? Type 1 has only one recognition site. Okay, one recognition site. So when they recognize only one site, okay, so that means their application is highly limited. Okay, so we need multiple recognition sequences. Only then we will be able to edit a very large a large varieties of sequences, and we can go for different organisms for genetic editing. Whereas when it comes to type 3 restriction enzymes, type 3 restrictions enzyme have their own limitations, where these restriction enzyme, imagine that you have a gene sequence from here to here. So imagine a restriction enzyme cutting away, very far away from the gene sequence. So that means you are carrying unwanted load. Now, when we are doing a genetic engineering, we don't want any unwanted gene, right? Unwanted nucleotides. We want it very, very precise. So that precision comes with type 2 restriction enzymes. So type 2 restriction enzymes bind to a very, very specific recognition sequences, and they cleave 2 points on the DNA strand. So that is why it is highly applicable when it comes to recombinant DNA technology. So now, when we talked about nomenclature of restriction enzymes, right? So while we're talking about nomenclature of restriction enzyme, we took a couple of examples like EcoR1, HIN2 or HIN3 or BAMH1. So these are the some of the restriction enzymes we use on a daily basis for genetic engineering. So now, when we talk about the recognition sequence, I told you that restriction enzymes have the capacity to recognize the repetition of sequences, like 3 sequences in a repetition, or 4 sequence, or 5 sequence, or 6 sequence. So we call it as trimer, tetramer, pentamer, and hexamer sequences. So based on their recognition capabilities, we can even predict how big will be the fragments that they cut. For example, in trimer sequences, I told you there are 4 nucleotides, so there is repetition of 3 times, right? Trimer means 4 4s are 16, 16 4s are 64. In tetramer, it will be 4 power 4. In pentamer, it will be 4 power 5. Or in hexamer, it will be 4 power 6. Okay, you get my point. So what does these numbers indicate? These numbers indicate that if I have a DNA strand and I apply a restriction enzyme that can recognize a trimer sequence, that means I can predict for every 64 sequence there might be one cut. That means my DNA strand that will be cut by the restriction enzyme will be almost up to 64 base pairs long. So this will give us an ability to identify, okay, they have this kind of recognition sequence, and this will be the length of the fragments they might cut. Got it? So one more important aspect is what type of recognition sequence do these restriction enzymes have. So we call them as palindromic sequences. So whenever we write about palindromic sequences, the most common example that is given in your textbook is Malayalam, right? So, or see, Malayam, LAM. So when we talk about Malayalam, so 5 prime to 3 prime we read, and we will also tell that we will read from again 3 prime to 5 prime. So that means the spelling is same in both the ends. But where we confuse is, we confuse that we are reading in only one frame. So, but if you look at the DNA is a double-stranded sequence, isn't it? For example, if I take eco R1, look at the recognition sequence. If I tell that it is GAA, okay, GAA-TTC is a recognition sequence here. Okay, you look at this 5 prime to 3 prime. When I read, I will go like this: GAA-TTC is a recognition sequence for eco R1. All right. Now go from 3 prime to 5 prime. It will go like this. See, you can come here, or CTTAAG. So this is how we read a palindromic sequence. So if you look at eco R1, it is hexamer sequences. Similarly, if you look at hint 3, okay, look at hint 3, you can go like this: AGCT is a sequence, isn't it? Look at here, AGCT. So in both 5 prime to 3 prime direction, when you read, it will mean the same. The spellings are same. So these sequences are called as palindromic sequences. Understood? So these palindromic sequences are a specific recognition sequences that these restriction enzymes will go and attach to, and they will cleave in these regions. Understood? So let us see. There are some important examples for palindromic sequences. For example, it is given GAA-TTC. So where do we find that GAA-TTC? So you can see here, GAA-TTC is a recognition sequence for eco R1. So when it comes to hint 3, look at hint 3 sequence is completely different. AAGCTT, AAGCTT. So each of our restriction enzyme have their own unique recognition sequence. But one thing is common in all of this restriction enzyme, that they have the capabilities to recognize palindromic sequences, mostly palindromic sequences. Understood? So this is about it. So when you talk about the action of restriction enzyme, based on their capacity to cleave on both the strands, they will result in a specific type of ends. Okay, when we talk about specific type of ends, let's come back to this slide. Okay, so you can see here, you can see the red color arrow marks that are present on this slide, right? So you can see when you talk about BAM-H1, okay, and you talk about ALU1, okay, ALU1. So you can see here, in this case, the cleaving is happening on both the strands at a same site. When the cleaving happens at the same site, that will result in ends like this. So these ends, okay, for example, if you cut a wood very equally, that is called, these ends are called as blunt ends, okay. Whereas when you look at BAM-H1, look at where they are cleaving. So, it is cleaving at one side, and this is cleaving at the different side. So that will result in ends like this. Okay, so hold on, sorry. Something like this. Yeah. Now you can see the difference between the two ends. This is called as blunt end, and this is called as sticky. Okay, sticky ends. So now what is the purpose of having this blunt ends or sticky ends? So when you use when you try to genetically engineer, sometimes we don't use only one type of restriction enzyme. We end up using many restriction enzymes in the same engineering process. So during this process, what happens if you are not aware about what type of ends we are creating? Then it will be very difficult for us to stitch back together, because we are not just cutting it, we are engineering it to stick back together to the, for example, if you take plasmid, if I take a plasmid DNA, so if I take a plasmid DNA and if I want to engineer this plasmid DNA into a recombinant plasmid, so I want to cut it here, right? So imagine that I want to make more alterations in cutting at several sites. So when I want to cut it, I want to put it back together in its original shape. That is when it will be capable of self-replication and expression and all. So if that doesn't happen, then all of this process is waste. So that is why what type of restriction enzyme we use depends upon what type of ends it creates. Okay, so usually our scientists will go for the similar type of restriction enzymes giving rise to similar ends, either sticky ends or blunt ends. But mostly we choose sticky ends. Why? Because during the ligation process, it's very easy for us to create these complements and stick their DNAs together. So you can see here how neatly this ends will work. Okay. So this is the example for how the restriction enzymes actually act and what type of ends they create based on their action. Okay. Fine. We understood what is sticky ends and what are blunt ends, correct? So fine. So we understood what is restriction enzymes, and now what is the types of restriction enzymes and what is the action of restriction enzymes, right? Now, why do we need restriction enzymes? You already know. Restriction enzymes are required to cut the DNA at specific location so that a recombinant DNA can be attached to that particular vector, so that it can be put in the host. So that means restriction enzymes have a very crucial role when it comes to genetic engineering. As simple as that, correct? So now once we create a recombinant vector, okay, once we create a recombinant plasmid we have chosen, right? So once we have created that, then why did we actually insert our gene of interest into our plasmid DNA? We call this plasmid DNA as a vector or a vehicle which helps in carrying our gene of interest from from us to the host, right? So now we are talking about cloning vectors. The second tool of this genetic engineering is called as cloning vector. Now, what are these cloning vector? As I said, cloning vectors are the vehicles that carry our recombinant DNA or foreign DNA into the host cell. So once they do that, okay, that means one part of our genetic engineering is complete. So examples of these cloning vectors are plasmid DNA, as we discussed, and also I mentioned about viral DNA. You can talk about bacteriophage. We call it as phage vectors. Okay, or you can also call it as retroviral vectors. So etc. So there are many types of vectors. Now, for a vector to be ideal for us to use it, they have to have a specific characteristic features. Only then we can use a vector. Okay. So first one is presence of origin site. Okay, we'll just mark down all the important characteristic features of a cloning vector. Then we will discuss one by one what are the significance with respect to individual headlines. Okay, so first one, what is the characteristic feature we need? We need origin of replication. Second one we need is a selectable marker. Third one is a requirement of a cloning site. And fourth one is vectors for cloning genes in plants and animals. So this one is different. Okay, first 3 points are very important when it comes to features required to cloning vector. Okay, so let us talk about origin site. See, why do we require origin site or ORI site? When it comes to ORI site is a site where your the initiation of replication of DNA takes place. That means if the DNA has to be made more copies, then it starts with ORI site. Now, without the origin of replication, your vector cannot create more and more copies of your foreign DNA, all right, or the recombinant DNA. So if you cannot make more and more copies of our target DNA or recombinant DNA, then there is no use of genetic engineering. Now, why are we specifically talking about origin of replication? Now, every plasmid DNA initially before being engineered, they will have the ORI site. Okay, see, for example, what I'm trying to say is this is your plasmid DNA before your engineering process. Now, let us say that here is your ORI region. Okay, ORI site. Now, what will I do? For me, I want to insert this gene of interest into this vector, that is plasmid vector. During this process, what will I do? I will use restriction enzymes, correct? I will use RE. When I use restriction enzyme, my restriction enzyme will cleave at several sequences or several sites. Usually we want to do it at only two locations, but sometimes we end up having many small fragments. Okay, when we have that, so now what happened? Our expected site at which your recombinant DNA will occupy was like this. Okay, let us say that from here to here, my gene of interest will occupy. So when my gene of interest will occupy, then ORI site is unaffected. It is normal. It is in the normal position. Our gene of interest is also in the normal position. Then there is no problem. But as I said, when we cut the plasmid DNA for the insertion of foreign DNA, we cannot actually control this process of ligation. When I tell this control the process of ligation means after we have completed this introduction of our foreign DNA into the vector which is treated by the restriction enzyme. Now when we are giving ligase, now when the pieces come back together, we cannot predict how it's going to reorganize itself. So during the reorganization process, now as I said, you have your ORI site. Okay, imagine that your recombinant DNA or the foreign DNA occupies the position of your ORI site. So that means what? Your plasmid or your vector that is carrying your foreign DNA will be able to express, correct? It will be able to express, but will it be able to make copies? Is it not? It's not possible, right? So when it cannot make copies, then is there any use of this vector? Definitely no, right? So we can have it for one generation, but when you go for a prolonged cycles of cell, then you cannot produce the same type of cell with the same clone characteristics. So that is why origin of replication is very, very important characteristic feature for a cloning vector. Understood? Okay. Next one is a selectable marker. What is a selectable marker? See, selectable marker can be anything that will help us to distinguish between what are transformants and what are non-transformants. That means which of our vectors have my foreign DNA and which of the following vectors do not have my foreign DNA. So whichever have my foreign DNA are called as transformants, whichever do not have my foreign DNA are called as non-transformants. So this ability of identifying, okay, this it will give us an a platform or a tool to identify which is transformed and which is non-transformed. So that tool is called as a selectable marker. So where can we find a selectable marker? Okay, let us take an example. So selectable marker can be, so for example, if we take a plasmid DNA, we choose selectable markers as antibiotic resistance gene. Now let us break down this. What is antibiotic resistance gene? See, we know that for any gene that has to be given value or signified, it has to express. And we know that in E. coli, apart from the nuclear DNA, see, this is your nuclear DNA, whatever is present here, that is in the E. coli. Outside this nucleus, what do we have? We have a double-stranded circular DNA called as plasmid DNA. Why do we even consider this plasmid DNA? And why do we give it important? We give it important because it controls some of the characteristics of the cell. That is why we signify this particular DNA, even though it is extra chromosomal DNA. So what are these characteristics that it gives to the cell? One of the important abilities that the cell gets is antibiotic resistance. Now, we use antibiotics for several diseases, correct? So whenever we get a bacterial infection, whenever we get a fungal infection, so whenever we get a protozoan infection, we use antibiotics. What is antibiotics? Anything that kills these microorganisms are called as antibiotics, which are naturally produced. So if you see your environment, as I said, so there is a lot of interaction between organisms, right? So bacteria will interact with fungi will interact with viruses. So all of these organisms are actually attacking each other, okay, or predating on each other. So during this process, what happens? Let us take that a fungi is there and a fungi is being infected by a bacteria for several generations. So if the fungi keeps quiet without adapting, then eventually it will go extinct. So that is why the fungi will build a defense mechanism. What does it build? It will try to produce some toxins against this particular bacteria. And these toxins are nothing but antibiotics, correct? No. So now this bacteria, whenever it enters the fungi, because of the production of these antibiotics, it will die. But how long can it happen continuously? Until the bacteria will gain the capacity to adapt to that antibiotic. So as it keep on exposing to these antibiotics, now the bacteria will gain resistance towards it. So this ability to resist the action of antibiotics, okay, comes from antibiotic resistance gene, which is present in the plasmid DNA. Okay, so we use this gene, which is present in the plasmid DNA, as one of our selectable marker. Now, why do we use that? Because in the plasmid DNA, if we analyze, there are several selectable markers in the form of antibiotic resistance gene. For example, ampicillin is an antibiotic, so ampicillin resistance gene. Okay, So you have chloramphenicol resistance gene, tetracycline resistance gene, kanamycin resistance gene. So these are the resistance gene. Now what do? We try to insert our gene of interest in the place of this, resistance gene. Did you understand? So for example, if a cell is able to see tetra means tetracycline resistance. So if I apply my gene of interest at the place of tetracycline resistance, what happens to the cell? If I grow this recombinant cell, okay, or transformed cell on a tetracycline medium, will it grow? Definitely no, right? It doesn't grow because it will die. Why it will die? Because whatever the gene that was there that was giving the capacity to resist against tetracycline, now it will not have that capabilities, right? So now we can differentiate if the cell is transformed or non-transformed. If it grows in tetracycline, that means that it is not having the foreign gene. If it does not grow in the tetracycline, that means that it has the recombinant gene. So these markers which help us in distinguishing between transformants and non-transformants are called as selectable markers. Okay. Okay, students, are you understanding? So do you have any doubts? Give me a second. I'll just check on the YouTube, students. YouTube students, if you have any doubts, you can just post your doubts on the chat box. All right. So I'll be looking at the chat box. At least at the end of the session, I will see what's happening in the chat box. All right. So we can solve our doubts. Okay. Okay, let's get back to the session. Fine. Did you understand what is selectable markers and the significance of selectable markers? Okay. Now the next one we have to talk about is cloning sites. So what is a cloning site? Usually when we talk about insertion of alien DNA, so where do we insert this alien DNA onto our vector? Where can we put our alien DNA or foreign DNA into this vector? So usually these recognition sequence which allow us to insert our gene of interest are called as recognition sites for restriction enzymes. So based on the choice of restriction enzyme that we make, see, for example, if I take a plasmid DNA again, okay, so imagine that here is my selectable marker and I will choose a specific restriction enzyme. I will choose a restriction enzyme which can cleave as close as possible to this selectable marker so that this site is empty for my cloning. That means I can put my gene of interest at this location, correct? No. So now if I do not have a recognition sequence anywhere near my selectable marker, now it will become a very difficult task. Why it will become a very difficult task? I will have to look for the other restriction enzyme which is very nearest to my selectable markers. So these regions which allow us to use the restriction enzymes and create this site for insertion of our alien DNA, these are called as cloning sites. Understood? So usually we prefer that there is a single recognition site when it comes to a vector, but eventually it will end up having several recognition sequences. Whenever we have several recognition sequence, then it will be multiple fragments. I was talking about how the multiple fragments happen in the plasmid. So it will result in formation of multiple fragments. When we have multiple fragments, then stitching them back together and forming a functional vector will be a difficult task. Understood? So once we have a cloning site, once we recognize the cloning site, we use the specific restriction enzyme, we insert our DNA, and the insertion of DNA happens through ligation, correct? No, we provide ligase, an enzyme, where our alien DNA is carried out to this restriction site or cloning site, and then it will be stuck together. I mean, like it will be attached at some position where we wanted it to be present. Understood? So these are about cloning sites. Fine. So now they have given an example, correct? No. So when we talk about ligation of foreign DNA, correct? No. See, there are given two examples. Just we will take these examples. BAMH1 is one of the vectors. So in BAMH1, tetracycline resistance gene, okay, in vector PBR322 is used as the site, okay, for the insertion of our recombinant DNA. Okay, If the ligation do not happen at this site, then it is called as non-recombinant plasmids. Did you understand? Or they do not carry this specific character of our foreign gene. Understood? So when they have this exact gene at a tetracycline resistance gene, then we call it as a recombinant plasmid. When they do not carry this specific character at a specific location, we call it as non-recombinant plasmid. Understood? So now we know what are the tools that we require for creating a recombinant vector, genetic engineering. Okay, to how to do genetic engineering, and what are the tools that we need for genetic engineering. Correct. Now, next we'll go for how do we select the cells that are transformed or not. Okay. So fine. What do we do now? Now we know that we have inserted our foreign DNA at a selectable marker. Okay, we have applied them on a selectable marker, and that marker will be our antibiotic resistance gene. At an antibiotic resistance gene, what have I done? See, for example, if I have a plasmid like this, okay, let us say that there is a tetracycline resistance gene. I will write TET, TET-R. So at TET-R location, what I have done, I have inserted my gene of interest. Correct? No, this is what I wanted to do. When I do this, my vector has become recombinant vector. Correct? No. So this process where I have inserted my gene of interest, okay, in the place of an antibiotic resistance gene. So this process is called as insertional inactivation. What did I do in this? When I inserted my gene of interest in the place of an antibiotic resistance gene, so the antibiotic resistance character of that particular plasmid is inactivated. So this is a methodology for me to understand whether it is recombinant or non-recombinant, or transformant or non-transformant. Did you understand? Now here what happened, whenever I insert a gene in the place of tetracycline resistance gene, as I said, what will happen to this recombinant plasmid? They will lose tetracycline resistance due to the insertion of foreign DNA. Did you understand? So let us see what is happening here. Now I have a transformed cell. I do not have a transformed cell. Now I will grow these cells on a medium containing amphicoline. Understood? So now when I grow a cell containing on amphicoline, now what will happen? Only the cells that have amphicoline containing media will grow. Okay. Whereas the cells which do not have amphicoline will not grow. Correct, isn't it? See, I will give you one more example. It is even better. Okay. See, now they have talked about, for example, if I take one of the plasmid DNA, and the plasmid DNA have tetracycline. Okay, TET, I'll write it tetracycline, and another antibiotic is amphicoline. Okay, amphicoline resistance gene. Now what happens if I do insertional inactivation? That means my tetracycline character is knocked out. Understood? So now I will grow my cells in a medium containing amphicoline. If I grow my cells medium containing amphicoline, that means my recombinant cell should have to grow on amphicoline containing media. Correct? No? Whereas if I grow my if I grow my recombinants on tetracycline containing media, will they grow on tetracycline containing media? No, definitely no. Why? Because they have been inactivated, because they do not have tetracycline resistance gene. So these cells which cannot grow on the tetracycline media but can grow on amphicoline media are called as transformants. Did you understand? Okay. What are non-transformants? Non-transformants, what they will do is they will see, for example, you can take it here. Hold on. See, non-transformant, they have no plasmids, so they do not resist either tetracycline or amphicoline. You can see here. When you talk about non-transformed cell, that means they have not taken any of the vectors into their cell. So when they do not take any of the vectors into the cell, that means that these cells do not have a capacity to resist either amphicoline or tetracycline. Did you understand? And the third variety is transformant with recombinant plasmid. So whenever we talk about transformants with recombinant plasmid, they can resist only amphicoline, and these are the perfect vectors. When we talk about transformant with non-recombinant a plasmid, they resist both tetracycline and amphoxylin. Why they are resisting both tetracycline and amphoxylin? See, here is your vector and here is your tetracycline, here is your amphoxylin. Now the gene of interest has happened here, insertion has happened here. That means what? It can resist both tetracycline as well as amphoxylin, as well as it has the characteristic of a foreign gene. Understood? So these are called as transformants with non-recombinant plasmid. And here, the ones which can grow only on one media, that is amphoxylin media, not the tetracycline media, these are called as transformants with recombinant plasmid. Understood? And non-transformant cells are the one which do not have any vector at all, which do not take, which have not taken any vector at all. Okay. So these are the three types of cells we get through insertional inactivation. Remember that. Okay. Okay, students, you have any doubts? If you have any doubts with respect to transformants, non-transformants, recombinants, or insertional inactivation, you can ask me. Okay. I'll give you a minute. So you can just think about it and you can try to answer. What about students on the YouTube? So even for you, it's the same thing. If you feel anywhere that you have missed out or you are not able to get it, you please let me know so we can continue our session. So is it clear, people? Please interact at least at one point. Okay, it will be helpful for all of us. Fine. So we talked about insertional inactivation, isn't it? But when it comes to insertional inactivation, sometimes it is very difficult to screen exactly the recombinants from non-recombinants. It will be a very difficult process and sometimes it is a failure also. So that is why scientists developed an alternate selectable markers. So this selectable markers was very, very elaborate. Okay, it's very interesting. Why it is very interesting, I will tell you. So when it comes to insertional inactivation, when there is a misposition of our foreign gene, okay, then we can expect that there is a possibility that for us to screen transformants and non-recombinants, and transformants and recombinants will be little bit tedious task. Okay. So that is why they wanted to go for another method where there is strict transformants, okay, and non-recombinants. So these cells we wanted. So how do we do that? For that, our scientists will choose one of the very beautiful gene, and this gene is called as, your lac gene. Correct? No, the lac gene in the bacteria has how many parts? We know that they are having 3 parts. Correct? No. What are the 3 parts? We have lac Z, then we have lac A, and then we have lac Y. Correct? No. For us, our gene of interest is lac Z gene. Okay. Why lac Z gene? It will produce something called as beta galactosidase, and beta galactosidase helps in breaking down your lactose into glucose and galactose. Correct? No, people. So this is what we have learned from gene expressions in prokaryotes. Correct? Excuse me. So why did we choose lac Z gene specifically as a selectable marker? See, the characteristics of this lac gene is it is highly mutagenic. What is the highly mutagenic means? For example, see, this is the gene, right? For example, let me tell you something. This is the gene. Let us assume that this is the gene. Now, let us say that I have chosen lac Z gene as my marker. That means I have to put my gene of interest at the position of lac Z gene. During this process, what happens? Okay, assume that my gene of interest occupies from this position to this position. Instead of from here to here, I'll do it like this. Okay, from this position to this position, it has occupied. Then a small portion of your lac Z gene is left out. Even though there is a very small amount of your lac Z gene that is left out, still the lac Z gene can produce a functional beta galactosidase. Understood? That is the catch here. So when it produces functional beta galactosidase, that means it can still break down lactose into glucose and galactose, correct? So when can we completely deactivate our LAGZ gene? Correct. So we can completely deactivate LAGZ gene when we have total insertional inactivation. What is total insertional inactivation? When my gene of interest occupies completely over LAGZ gene without leaving a little bit also, then I have complete inactivation of my LAGZ gene. Did you understand why did we choose LAGZ as a selectable marker? Now, what are we doing? Now we will grow these cells in a medium containing lactose, but not lactose. So we have another substance called as analog of lactose, we will call it as. Okay. So we will use an analog of lactose. Why do we use analog of lactose? Okay. See, analog of lactose that we use in this medium is called as XGAL medium. Okay. So what is XGAL medium? It is similar to lactose, but it is not lactose. Okay, very similar to lactose. Whenever a beta galactosidase will act on XGAL, it will break down. When it breaks down, it will produce a chromogenic substance in the form of a precipitate, which is identical to blue color. Okay, see here, it will give rise to blue color. Did you understand? Now what happens, people? Let's see what happens. I have put my foreign gene, correct? No, foreign DNA, and I have put it at the LAGZ position. Here there is no proper insertion on the LAGZ position. Now what happens if in this case, if my gene of interest is here or LAGZ is here and my gene of interest has occupied completely here, will it produces beta gal? No production of beta gal. When it does not produce beta gal, what kind of colonies that it produces? White color colonies it will produce. Okay, white color cells or colonies. Whereas when you have, even though there is, for example, this is your what do you tell, your LAGZ gene. Let us say that your gene of interest is here, okay, or here. So only a little bit portion of your LAGZ gene is left out. Even though there is a little bit portion of your LAGZ gene left out, what did I tell? It will produce what? Beta gal is produced, right? When beta gal is produced, the XGAL medium will be broken down and the precipitate will be formed in the form of blue color. So we will get what color colonies? Blue color colonies. Now, is it easy for me to recognize which is recombinant, which is transformed, and which is non-transformed? All the white colonies are my cells. All the blue colonies are the ones that I want to neglect. Now, what do I do? I'll just scrape these white colonies and I'll grow them in a very huge batch so that eliminating all the blue cells. Can it happen? So now directly I've eliminated a big headache of having the difficulty of identifying which is recombinant, which is non-recombinant, and all that things. Now I have a perfect cells which are transformants and non-recombinant cells in a one go. So this is one of the wonderful tools that we have developed, the scientists who have developed to recognize, which are the cells that really carry our gene of interest at exact locations. Okay. So after this, it helped a lot in creating more genetically engineered products with high precision rates. Okay. So fine, people. So we have 2 minutes left. I think we will end the sessions. So before I end the session, as usual, I will put a poll on NPS. So please try to answer. I mean, like, react to the NPS because the interaction is quite low. So I would expect you to answer even if you don't know. Try to respond, okay? Or ask doubts. Something should happen. Okay, please help me fill this form because it is necessary. Okay, students, thank you for your response and have a nice day. I'll meet you in the next session and we'll discuss the rest of the topics and see you soon. Bye bye.